For decades, we have been told a simple and compelling story about our place in the natural world. It is the story that we, as humans, share an astonishing 99% of our DNA with our closest living relatives, the chimpanzees. This single percentage point has been used to explain so much, suggesting that the vast differences we see between our species, our complex languages, our sprawling cities, our unique consciousness, all spring from a tiny sliver of genetic code. It's a powerful idea, one that has shaped our understanding of evolution and our own identity for a very long time. This figure, however, is beginning to look like an oversimplification, a remnant of an earlier era of genetic science. The 99% claim was born from methods that, while groundbreaking at the time, could only see part of the picture. Imagine trying to compare two enormous, intricate encyclopedias by only looking at the pages that had nearly identical paragraphs. You would get a sense of their similarity, of course, but you would miss all the unique chapters, the rearranged sections, and the pages that exist in one book but not the other. This is precisely what early genome comparisons did. The original studies focused on the most similar, easily alignable parts of our genetic code, specifically the protein coding genes. These are the parts of DNA that provide the direct instructions for building the proteins that make up our bodies. By lining up these specific sequences from a human and a chimpanzee, scientists could count the differences, which were indeed few. This method, however, deliberately ignored the vast, complex, and structurally different regions of the genome that were simply too difficult to compare with the technology available at the time. New research, including a landmark study published in Nature in early 2025, has dramatically changed this perspective. Using advanced techniques that allow for a complete end-to-end comparison of entire genomes, scientists are now able to see the full story. This more comprehensive view includes vast stretches of DNA that were previously left out of the equation. When these once-ignored regions are included, the picture of our genetic relationship with chimpanzees becomes far more nuanced, and the 1% difference starts to look like a significant underestimate of the real genetic distance between us. To understand why the old number was misleading, we need to think about what DNA actually is. Our genome is often described as the Book of Life, written in a four-letter alphabet, A, T, C, and G. These are the chemical bases that form the rungs of the DNA ladder. The human genome contains about 3 billion of these letters, all arranged in a specific sequence. Comparing our genome to a chimpanzee's is not just about looking for simple spelling differences or single-letter changes in an otherwise identical text. It is far more complex than that. A more accurate analogy would be to compare two versions of an epic novel. The original 99% figure was like finding that both books use the same words and have many identical sentences. But the new, more complete comparisons are like discovering that one book has entire paragraphs, pages, or even whole chapters that are missing from the other. Or perhaps one book has a chapter that has been copied 3 times, while the other has it only once. These larger-scale differences are called insertions, deletions, and rearrangements, and they are abundant. These structural variations are not trivial. A single deletion could remove a vital instruction, while an insertion could add a new one. A large chunk of DNA being moved from one chromosome to another can completely change how the genes within it are controlled and utilized. The 2025 study revealed that these structural differences are far more common than previously thought. When you add up all these missing and rearranged sections, the parts of the human genome that have no direct or alignable counterpart in the chimpanzee genome amount to a substantial portion of our DNA. This is why the number has shifted so dramatically. If you only compare the parts that are directly alignable, the protein coding genes, you get a number close to 99% similarity. But this ignores about 15 to 20% of the genome that cannot be so easily matched up. When scientists factor in these huge structural differences, the overall genetic similarity drops significantly. The latest estimates, based on this whole genome approach, now suggest the real difference is closer to 14 or 15%, a figure more than 10 times larger than the one we have become so accustomed to hearing. For a long time, scientists believed that the vast majority of our DNA, around 98% of it, was essentially useless. They called it junk DNA because it did not appear to code for proteins, which were seen as the only important product of our genes. We now know that this view was profoundly mistaken. This non-coding DNA is not junk at all. Instead, it is a vast and complex regulatory system, acting like the conductor of a genetic orchestra. It tells our genes when to turn on, when to turn off, and how loudly to play their part in the symphony of life. These regulatory regions are the key to understanding how small genetic changes can lead to enormous physical and behavioral differences. The genes themselves might be very similar between humans and chimps. We both have the genes for building a brain, a hand, or a larynx. However, the non-coding DNA that controls these genes is vastly different. It is like two chefs having the exact same set of ingredients but using entirely different recipes. One recipe might call for a dash of an ingredient, while another calls for a cup, leading to two completely different dishes. Think, for example, of brain development. The genes that build the basic components of a neuron might be nearly identical in humans and chimps, but tiny changes in the regulatory DNA can alter the timing of when these genes are active during fetal development. A gene that stays on for a little longer in a human fetus could lead to a massive expansion of the cerebral cortex, the part of the brain responsible for higher thought, language, and abstract reasoning. This is how evolution can produce such dramatic outcomes without completely rewriting the genetic source code. This is where most of the newly discovered differences between our species lie. They are not in the protein coding genes themselves, but in the vast expanses of regulatory DNA that surround them. These differences in the genetic switches have a ripple effect, changing the way our bodies are built and how they function. A subtle change in a regulatory sequence can alter the expression of dozens of genes, leading to the profound differences in cognition, communication, and physical abilities that so clearly separate us from our chimpanzee cousins. The discovery that we are perhaps only 85% genetically similar to chimpanzees does not overturn the fundamental truth of evolution. Chimpanzees are still our closest living relatives in the animal kingdom, and we undoubtedly share a common ancestor who lived millions of years ago. What this new understanding does, however, is provide a genetic explanation that better aligns with the reality we observe. The old 1% figure made the vast chasm between our species seem almost miraculous, a biological puzzle that was difficult to explain with such a tiny genetic toolkit. A larger genetic gap makes much more sense. It helps us understand why humans can compose symphonies, develop calculus, and build global civilizations while chimpanzees, for all their intelligence and complexity, cannot. The significant differences in our non-coding regulatory DNA provide a powerful mechanism for generating the unique traits that define our humanity. It emphasizes that evolution is not just about changing the genes themselves, but also about rewiring the intricate network that controls them, allowing for new and complex features to emerge from an ancient set of genetic parts. Furthermore, this new perspective reminds us of the complexity within other species. The same 2025 study highlighted that different populations of chimpanzees can have a genetic variability of up to 9% among themselves. This is a staggering amount of diversity compared to humans, who share about 99.9% of their DNA with each other regardless of where they live on the planet. Our species is remarkably uniform genetically, while our closest relatives show incredible variation. This insight adds another layer to the fascinating story of primate evolution and our own unique evolutionary path. In the end, the story of human and chimpanzee DNA is not one of simple percentages. It is a tale of complexity, of structural change, and of the profound importance of genetic regulation. While we remain closely related to chimpanzees, the differences in our DNA are far greater and more meaningful than we once believed. This deeper understanding does not diminish our connection to the rest of the natural world. Instead, it enriches it, giving us a more realistic and scientifically robust foundation for what makes our species, for all its flaws and wonders, truly unique.